Now there are two main types of fats, saturated fats and unsaturated fats. Saturated fats have as many hydrogens as possible bonded to their carbons. Unsaturated, and so they end up with long straight chains. Unsaturated fats have double or triple bonds between some of the carbons, so they wind up looking kinky. Literally, kink, kink, kink. So this here, the stearic acid, is a saturated fat. It has all single bonds. This oleic acid here is unsaturated because it has a kink, and this linoleic acid has many kinks to it. A triglyceride, which is something that you hear about, especially in relation to blood tests, is glycerol, which is this puppy up here, bounded to 3 fatty acids. So it's one thing here with 3 tails. And these are found in your bloodstream. You can break them down for energy, or they can just clog your arteries. Now, you probably have heard a lot in the news about trans fats. I know some cities have been banning trans fats. Trans fats are different from regular fats in how the hydrogens are arranged around the double bond. So these are unsaturated fats, but they're different than normal ones. And these have been found to raise cholesterol. And the reason why this happens is in nature, there aren't any trans fats. The natural trans fats are made artificially when you add extra hydrogen to vegetable oil to make it solid at room temperature. So this is a chemical process. This is why instead of just having butter, now we have like parquet. So here's a picture of that. This is what it's supposed to look like in nature. This is what it does look like in the lab. And for some reason, your body can, even though looking at that, you probably couldn't tell the difference, your body can tell if you're eating one or the other. Phospholipids are going to become very important later this unit when we talk about cell membranes. Phospholipids have a phosphate head. See the P right there? And this head here is hydrophilic. It's water-loving. It's because it has charges on it. It has a negative here and a positive over here. The tail is made out of fat. The tail is very hydrophobic. So you're going to have one end of this thing that likes water and one end that doesn't like water. And that's how you get the cell membranes. So here's the part that likes water, here's the part that doesn't like water. If you put some phospholipids in a beaker of water, they will spontaneously form what's a cell membrane. So they form in 2 layers, and they're standing with their tails together so that there's no water by their tails, and their heads are facing the water. So you end up with this double layered membrane where you've got the outside there and the inside there. Another group of lipids are the steroids. Steroids are lipids with a rigid backbone of 4 carbon rings and no fatty acid tails. These are used by your body as signaling things. This is testosterone. Over here you have cholesterol. Okay, now we're on group 3 out of 4, the proteins, like that juicy steak. Proteins are polymers made up of monomers known as amino acids. Proteins make up the structure of animals. So you are mostly water, and after you take the water out, you're mostly protein. And there's one special kind of protein we'll talk about called enzymes. Enzymes are special types of protein that make reactions in a cell happen. So here's how an amino acid, one of these monomers that proteins are made of, look like. They've got an N here and then 2 carbons. This is one end, this is the other end, and then this is the important part, this R here. There are 20 different things. Wow, what planet am I on? 20 different things that can go where that R is. And what you end up with, it's kind of like a charm bracelet. The chain here is this part. And then the charms are these R groups that are sticking off the side. Since you've got 20 different R groups you can put down in any order, proteins are amazingly versatile. That's why you can make skin out of protein, you can make hair out of protein, you can make muscle out of protein, you can make ligaments out of protein. There's so much of your body that's made out of protein, and so many different kinds of proteins. So here are pictures of the 20 different R groups. If you ever take biochemistry, you will be forced to memorize them. I really don't know why. It seems to be some sort of obscure rite of passage. Yeah. But anyway, for this class, you don't need to know them. Just know that there are 20 and that there are different kinds of them. They come in 4 main groups: things that are electrically charged, positive or minus, things that are hydrophobic, things that are polar, and then things that are so special we put them in their own group even though they're not related. So you can do a lot, and these side chains can have chemical reactions with each other and change the shape of the protein itself. So here's an example. Instead of just having an R here, we actually have the chemical formula for one of the side groups. This is valine. Now, the amino acids are joined together to form long chains called polypeptides. Guess what makes polypeptides? What kind of reaction makes amino acids turn into polypeptides? Here's a hint. That's right, it's a condensation reaction. So the first level of organization in a protein is its primary structure. This is the polypeptide chain. This is the order that you put the amino acids in. Then you have the secondary structure. The amino acids side chains, its R groups, will interact with each other, and you can form things like coils or helices and long sheets. Then you have individual coils and sheets interact with each other on the protein. So this is one long protein. Some parts of it have turned into sheets, some parts of it have turned into coils, and now you have a little pocket. This is kind of like a little catcher's mitt. This one, you had a bunch of different sheets that have formed together to form a barrel. And then you get into the quaternary structure, where you have 2 or more polypeptide chains that come together and make one big molecule. So the purpose of showing you these 4 different kinds of structures is to show you how complicated you can get with these things. They're not just a charm bracelet. You can make, so many complicated things in your body just using protein. Now, proteins can get messed up. Changes in pH or temperature can mess up proteins, and you guys will learn this when you do the enzyme lab. When proteins denature or get messed up, they lose their shape and they can't do what they're supposed to do. Okay, on to the last one, number 4 out of 4, nucleic acids. Nucleic acids have many functions. They can carry energy, they can help enzymes, and they can carry information. They can be DNA and RNA. So here is a nucleotide. This is one of the monomers in the nucleic acid polymers. Monomers and polymers again. So here is one nucleotide. You can see you've got phosphate groups over here. You've got a sugar here, and then this part here is the base. There are 4 different bases that you can have: adenine, cytosine, thymine, and guanine. And we'll see those again later, or on this slide. But they will be important later on in the course when we talk about genetics and that sort of thing. So adenines are A, guanine is G, thymine is T, and cytosine is C. And it's these bases pairing together that give you the instructions of DNA and RNA. ATP is something you may have heard of. It stands for adenosine triphosphate. So here's the adenine, the A. There's the phosphate groups. There's 3 of them. Adenosine triphosphate. Sorry about that. I keep hitting the tripod. So ATP is how cells store energy. When you break one of these phosphates off the sugar, it releases a bunch of energy, and your body can use this energy to do work. Now, RNA and DNA are two ways of storing, your cell can store information. RNA, or ribonucleic acid, assists in making proteins. So you use RNA as the instructions to make proteins. DNA, or deoxyribonucleic acid, stores genetic information. The two of them are identical, except for one oxygen. So here's the ribose sugar. Here it has an OH. Deoxyribose, it's literally without oxygen, deoxy. This hydrogen has lost its oxygen. Now this looks like just a little minor change, but it's actually a big deal. Deoxyribose is a lot more stable than ribose. So DNA can be used for long-term storage, while RNA really cannot. And here is the structure of DNA. You can see the double helix, you can see the phosphates and sugars are on the outside, and then the bases are in the middle. Now I believe this is my last slide. When you get to Lab 2, there are some pictures there on Lab 2 that show you what will be on the exam as far as the pictures you'll need to identify go. This is one of the pictures of DNA you'll have to identify. Since my exams are online and multiple choice, you don't have to, you won't have to draw DNA, but you will have to be able to recognize this picture of it. So look to the labs for the pictures that could be on the exam. Thank you.